Hi, we're doing Biology, Paper 1, AQA 2021 paper today. I'm going to do a walkthrough of this paper. I can start now. So, first question. A student prepared some animal cells to view using a microscope. Figure 1 shows the student preparing the cells. Name 2 pieces of lab equipment the student could have used to prepare cells to view using a microscope. Okay, so we want something that we can use to firstly get to the cell itself. What are we using here? It's an animal cell. And we also want to be able to see it, or basically just anything, any lab equipments. So there's a few of them that you can say. You can say the microscope slide. That's probably the most obvious one. Then we can also say some tweezers, scalpel, anything to take the animal cells. So you can say scalpel, tweezers. You can also say a swab. If it's an animal cell, you can get it from your cheeks. So you could use a swab. Tweezers, cover slip, that's the cover slip. Any dyes or stains. This is higher paper. Figure 2 shows the student's light microscope. Here we have part A. Wait, I'm just going to double check if this... Yeah, it's higher. AQA trilogy, yeah. Sorry. Sorry. So, we have name part A, eyepiece lens. That's correct. What is the function of part B? That's the focus. Yep. So, to focus the image. That's right. Clear resolution, focus the image, all of those are fine. The student tried to look at the cells using microscope. Suggest one reason why the student could not see any cells when looking through part A. No cells in field of view, yep. That's right, you can say that. You can say microscope is not focused, lens is dirty, mirror not in correct position, no cells in field of view, that's right. Bubbles in slides, yeah, you could possibly say that, yeah. You could say air bubbles, looking at air bubbles, that's fine, yeah. When will you be doing A-level bio or chem? I'll be doing both of them tomorrow. Chem will be in the morning at 11 a.m. and then bio will be in the afternoon around 1 p.m. This is AQA. Can you say low resolution? Yes, you can say low resolution is fine as well. Okay, let's have a look at the 6 marker here. Red blood cells are specialized animal cells. Compare the structure of a red blood cell with the structure of a plant cell. So you're required to know what the red blood cell does and what the plant cell does. So if you know what they are for, then you can kind of imagine how their structure will be. Red blood cell has no nucleus, that's correct. This is higher. This is triple bio, higher. So you can say red blood cells has no nucleus. Because it's an animal cell, it will have no cell wall as well. No chlorophyll or chloroplast. No vacuole. While the plant cells have everything else. So it has a nucleus, chloroplast, vacuole, and cell wall. That cell, I'm going to summarize it as RBC, biconcave shape. And contains hemoglobin. So we've talked about the differences here. Then we can talk about the similarities. I was going to write they, but technically you shouldn't write they. You can say both the red blood cell and the plant cell. Both red blood cell and plant cell have cytoplasm, cell membrane, and pigments. For the plant cell, it's green pigments. For the red blood cells, it's red pigments. Okay. So just by saying all of these, you get 6 marks already. You get 1 mark for saying the differences for each difference and for saying 2 similarities. When placed into a beaker of water, a red blood cell bursts, but a plant cell does not burst. Explain why the red blood cell bursts, but the plant cell does not burst. So, for this one, a red blood cell and a plant cell, you have to think about why we have a difference in them. So, for the red blood cell, we have cell membrane, but no cell wall. For the plant cell, they do have a cell wall. And something that we know about the plant cell is that the cell wall is what keeps them together. A red blood cell does contain a cell membrane. Red blood cell carries oxygen. Yes, that's right. It does carry oxygen, but it has nothing to do with why it bursts. So in terms of why it bursts, you can say, firstly, it's in a beaker of water, which means when you have the red blood cell, which looks something like this, and the plant cell, which looks something like that, when water enters, there's a lot of pressure that pushes everything outwards here. That's for the red blood cell. While in the plant cell, when the water enters, the outward pressure will be countered by the cell wall. So the cell wall will push it in. Therefore, it prevents it from bursting. So water enters the cell by osmosis. Plant cells has a cell wall which prevents it from bursting. Prevents the cell from bursting. For all exams, doesn't matter if it's bio, chemistry, physics, I need you to say that, or I need you to remember that you can never say he, she, they, we, it. Always specify what you're talking about. So if I say which prevents it from bursting, it's ambiguous. What is it? So you have to be more specific. You have to say this cell. It prevents this cell from bursting. Okay, number 2. A student investigated the effectiveness of 3 different antibiotics. Figure 3 shows how the student set up an agar plate. You have the bacteria here. Bacteria is growing on the agar, and you're testing each antibiotic on each disc. The student used aseptic techniques to make sure that only one type of bacterium was growing on the agar. Describe 2 aseptic techniques the student should have used. So, aseptic techniques meaning that we're trying to sterilize it. So that's one thing you can say. Sterilize the equipment before use. This means that when you apply a bacteria in it, only that bacteria will be present. No other ones will be present because it's already sterilized. So you can use sterilized agar. That's right. So you can use sterilize the equipment, use sterilized agar. You can say only lift the Petri dish a little so that no bacteria from the air comes in. Secure the lid of the Petri dish with tape. You can say that as well. Putting it upside down, that's interesting, but that would be allowed. Yes, that would be allowed. Because when you do it, you still have to say though, to do it like, to lift it up slowly. So you can say sterilize the equipment before use and use sterilized agar. Washing hands afterwards, that's not allowed. They don't want you to, they ignore washing hands. Ignore cleaning as well. You need to talk about sterilizing and securing the Petri dish. So if you were to add another one, secure lid of Petri dish with tape. This is because the air has bacteria that can enter. The student placed the agar plate in an incubator for 48 hours. Figure 4 shows the agar plate after 48 hours. Which antibiotic is the least? So remember here, we're looking at how the bacteria is growing on this agar plate. Originally, they all have bacteria, or at least supposed to have bacteria in all of them. But we want to see which one is the least effective, meaning the smallest radius, and that is B. So you can say has the smallest area where no bacteria were growing. Meaning kills the least bacteria. Smallest zone of inhibition is fine as well. Hi Zanera. So 2.3, calculate the area where no bacteria were growing for antibiotic C. Use pi equals 3.14, give the unit. Okay, so do we have any? Okay, so you're gonna need to measure this. I think I have a ruler. Oh yeah. I am on my iPad though, so it will be a little bit incorrect. That's around 1.1. Wait, what are we looking for? Antibiotic C. So that's around 1.8 for the diameter. Is that right? That's around 1.8. So that means the radius is around 0.9. So the radius is 0.9 centimeters. We're finding the area. Area is pi r squared. So that's going to be A equals pi times 0.9 squared. And that gives me a value. I don't have my calculator. So that's pi, 0.81 pi. And that would be in centimeters squared. You should calculate this out. You can leave it as centimeters squared, or you can convert this into millimeters first, which means you multiply by 10. So that would be 9 millimeters. Usually cells are measured in millimeters. So you can do pi times 9 squared. That would be 81 pi. Yes, this will be on my channel. No, you guys don't need to take away this part here, because technically this entire part is where there's no bacteria. This part here is where you have the septic already, right? So there will be no bacteria there anyways. So you don't need to take it away. Suggest one way the student could improve the investigation. So whenever you have the question improve the investigation, you can say repeat 3 times and take average, repeat and eliminate anomalies, use control disk, any of them is fine. Yes, this will be on my channel. If you guys don't know, my YouTube channel is, even I forgot, PCFG study. And I post my lives in there. So you can say here, repeat 3 times and take average. That's the best one, and that's the most common questions. Next one that we have, body mass index, BMI, is a way of finding out if a person's body mass falls within a healthy range for their height. Table 1 shows the information about 2 people, person A and person B. So we have the graph here, which is the BMI category of person A in table 1. Okay, let's have a look. So in table 1, the height in meters is 1.65. So that's over there. The body mass in kilograms is 63. So that's somewhere there. Is that somewhere there? Yeah, 63 is there. And that falls in the normal region. So they are normal. Calculate value X in table 1. Use the equation body mass index equals body mass over height squared. So we have BMI equals 92 over 1.71 squared. That is a value. I believe it should be around 31.5 kilogram per meter squared. Scientists think there is a link between BMI and life expectancy. Table 2 shows information about predicted life expectancy of men after the age of 50. Describe 2 patterns shown in table 2 about the effects of BMI category. Okay, so let's have a look at what we have here. They have describe, blah, 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 in table 2. Whenever you see the word describe, it always means they want you to actually give the values. So if you're saying it increases, you can't just say, okay, the BMI increase, but you need to actually specify it increased from a certain level to a certain level and give the values for it. So here we have normal is 19.06, living in bad health. The predicted number of years is 4.98. Okay. So overall, we can see that living in good health after the age of 50, for the normal, it's much higher than the rest of them, as compared to if you're overweight, obese, or clinically obese. So you can say that the normal, or the lower the BMI, the higher percentage or predicted number, the higher predicted number of years living in good health. For example, normal is 19.06, which is bigger than the rest. Question 2. No, not question 2. Part 2. Another pattern that we can see is that if you have going from normal to clinically obese in the living in bad health, there will be more of them at the higher BMI. The higher BMI shows higher years living in bad health. With the normal being below 5 years. There you go. The number of people who are obese in the UK is increasing. Explain the financial impact on the UK economy of an increasing number of people who are obese. Okay, so this one's kind of more of a logical question. They don't really expect you to, well, of course they expect you to know the answer, but they do give questions like these kind of to throw you out because you don't really learn about it. It's more like logical. You kind of gather the information from other subjects as well. So you can say a number of things. You can say cost of NHS, it costs the NHS more money, so they can't really treat everyone. There's more people who take time off of work. Like imagine COVID, for example. COVID had a huge financial impact, isn't it? So you can talk about that. Yeah, more people will have to go to the NHS, meaning more money will be spent. That's right. Cost the NHS more money as more people require treatment. A person who is obese is more at risk of arthritis. Arthritis is a condition that damages joints. Suggest how arthritis could affect a person's lifestyle. So, if you don't know the specific reason for why arthritis could affect someone, you can use something general. Firstly, we know that arthritis or any of types of these conditions could have a negative impact on the person's life. But think about how that impact would be. Again, this is more of the general one. Some of the impacts include losing job, losing income, mental health impact, things like that. That's more general. So you can say something general here. You can also say something specific if you do know what arthritis is. In this case, it would cause movement issues. And then, of course, you can say loss of income, mental health impact. These ones are more general, and you can use that for any diseases or conditions. A person who eats a diet high in saturated fat might become obese. Name 2 health conditions that might develop if a person eats a diet high in saturated fat. They can have coronary heart disease, so CHD. Type 2 diabetes, that's right. You can say any one of those: coronary heart disease, heart attacks, stroke, type 2 diabetes, cancer is possible as well. Hypertension, that's possible as well. Very good. Yeah, this test is pretty easy. This one's from 2021 as well, so it was recent. All living organisms respire. What is the chemical equation for aerobic respiration? So we know we want aerobic, meaning oxygen is present. And respiration is breakdown of glucose. So we just need to find the one that has oxygen and glucose in the reactants. And that would be the last one. That's right. (4.2), this is higher. 4.2, name the subcellular structures where aerobic respiration takes place. Yep, Ernie, you're way ahead of me. Everyone is ahead. Yep, that's correct. It's mitochondria. Powerhouse of the cell. Energy is released in respiration. Give 2 uses of the energy released in respiration. So what do you use your energy for? What was that? I think I saw movement. Yep, movement is one of them. I think someone said skeletal contractions, which is correct. So movement, keeping warm, active transport, growing. Actually, no, growing is not used. It's not included in this. That's more cell division. You can say cell division. You can say keeping warm because your skeleton will contract and relax, will shiver. Describe 2 differences between aerobic and anaerobic respiration in humans. Do not refer to oxygen. I'm using good notes for this. This is AQA. Anaerobic causes lactic acid. Yep, produces lactic acid. Anaerobic produces lactic acid, while aerobic does not. Aerobic produces energy. They both produce energy, but one produces less. Glucose breakdown by aerobic, they're both broken down. They can be broken down by either. I think someone said something about if they can talk about exercising, you can't talk about the exercise. So the best one is this one, produces lactic acid while aerobic does not. The other one is anaerobic, less energy. They both produce energy, just one is less than aerobic. You can also say aerobic produces water while anaerobic produces CO2. Aerobic happens in mitochondria, anaerobic happens in cytoplasm. You can say those as well. What are the 2 products of anaerobic respiration in plant cells? So we talked about this already. And I think someone did say the answer. What would be your answer here? Carbon dioxide and ethanol. That is correct. If you're saying the aerobic, then you would have the other one. You would have CO2 and water. A scientist investigated respiration and photosynthesis using some pondweed and a pond snail. Figure 6 shows the apparatus used. The apparatus was left in a well-lit room for 5 days. The data logger recorded the concentration of carbon dioxide continuously. After 5 days, the scientists completely covered the boiling tube with black paper, not black pepper. The data logger continued to record the concentration of carbon dioxide. Figure 7 shows the concentration of carbon dioxide inside the boiling tube over 15 days. Explain why the concentration of carbon dioxide in the tube stayed the same between day 0 and day 5. Okay, this one, you need to give values. Please make sure you actually input your values in here. So we're given a graph and they want us to talk about whatever's happening. So if they say use the diagram, use the figure, always put the values. Okay, explain why the concentration stayed the same between day 0 and day 5. Yeah, there's no exposure to light, so there's no photosynthesis. What was it? What was happening day 0, day 5? It's in a well-lit room for 5 days. And then what else happened? It's in a boiling tube, pond weed, pond water, pond snail. Okay. So for this one, you can say the pond weed takes in the CO2 that is released by the snail and pondweed during respiration. Yeah, there's both photosynthesis and respiration happening. So you can say rate of photosynthesis equals rate of respiration, for example. But you need to say that both the snail and the pondweed respire. It's not just the snail. Plants also respire. They do it at night usually. Suggest why the concentration of CO2 increased between day 5 and day 10. So after 5 days, the scientists completely covered the boiling tube with black paper. That means there's no more light. So there's no light present, so no photosynthesis. But of course, the respiration still happens. So when there's no light, no photosynthesis, and there's still respiration, then that means the rate of respiration increases while the photosynthesis stops. So there is a buildup of CO2 as snail and pondweed still respire. There you go. I think someone gave me a rose before. If you did, thank you very much. On day 10, the pond snail died. Explain why the death of the pond snail caused the concentration of CO2 to increase after day 10. So we have here day 10. And then we have an increase. And it's asking why we have that increase. Okay, so whenever anything dies, the carbon stores lock in the snail release, snail decompose. Yes, both of them. So the snail has a carbon store, and then when it gets decomposed, it gets released. So snail is being decayed or decomposed by decomposers. Therefore, CO2 is released during the decomposition or during the respiration of the decomposers. Respiration of the decomposers. You guys are so sweet. Thank you. Thank you for all the roses. Y'all, I do hope I win the Nobel Prize. That would be awesome. If you guys do want to win a Nobel Prize, by the way, there is one thing that my teacher told me. If you guys get to A-levels, or even now actually, in GCSEs, when you talk about the, organic chemistry, if you can figure out the number of isomers, I don't know if you guys know isomers yet actually, but if you know what an isomer is, and if you can figure out the number of isomers of, I think it was C18, you can win a Nobel Prize. Thank you so much, Connor, for all the roses. Okay, so next one that we have is amylase. Amylase is an enzyme that breaks down starch. Oh, let me just explain this one. So what is an isomer? An isomer is basically the same. So you know organic compounds, you can have a certain compound, let's say ethane. The ethane, you can have different forms of it. Well, not specifically ethane, but it could be anything else. So like propane, butane, all of them, they can have different shapes. They're all the same thing, they just have different shapes. That's what an isomer is. Okay, anyways, this one here. Name the type of smaller molecule. What did you guys say? Simple sugars. Amylase is a polymer of smaller molecules. Name the type of smaller molecules. Amylase is an enzyme that breaks down starch. Okay, so amylase itself is made of it's an enzyme, and all enzymes are proteins. So the monomer of it is amino acids. You guys are right. Name the 3 parts of the human digestive system that produces amylase. Pancreas, small intestine, salivary glands. Mouth, it's better to say salivary glands than mouth, but yes, all of them are right. So salivary glands, pancreas, small intestine. Explain how amylase breaks down starch. Answer in terms of the lock and key theory. Okay, so we have the lock and key now. This one, if they ever ask you this question, the answer is the same every single time. So you can just memorize it literally. So you have here, starch binds to active site. Starch is the substrate. Binds to the active site of the enzyme. Because shape of active site of the substrate and enzyme, well, the active site and the substrate is complementary. Are complementary. So fit together like a lock and a key. Chemical reactions occur to produce smaller molecules. Substrate binds to active site to make enzyme substrate complex. That's really good as well. I know this topic is really good. Yeah, to produce smaller molecules. That's right. You guys are all very good at this topic. A student investigated the effect of temperature on the activity of amylase. So figure 8 shows the apparatus used. This is the method used. Set up the apparatus as shown in figure 8. After 5 minutes, pour the starch solution into the amylase solution and mix. Remove 1 drop of the starch amylase mixture and place onto a spotting tile. Immediately add 2 drops of iodine solution. That's the starch test. To the starch amylase mixture on the spotting tile. Record the color of the iodine solution added to the starch amylase mixture. Repeat steps 3 to 5 every minute until the iodine solution stays yellow-brown. Repeat steps 1 to 6 using water at different temperatures. So we have here, the effective temperature is your independent variable, meaning that's the one you're changing. And then activity of amylase, that's your dependent. Name 2 control variables the student used in the investigation. So this one, you can't say temperature because we're saying here that temperature is what you're changing. A lot of the time, students think that the answer is temperature. That's because generally it would be, but in this case, it won't be. So you can say volume of, let's see what they used. Water at 5 degrees Celsius. You can say the temperature. Oh no, this one's already classified, so you can't talk about the water or the temperature there. You can say volume of the iodine solution. Volume of iodine solution used. Because if you use more or less, it will change the concentration. And then you can say volume of starch used as well. Volume of amylase, volume of starch, those are all fine. Temperature, you can say not temperature, sorry, the time before mixing the solutions, you can say that. Amount added to the spotting tile, so those things you can say. Why did the student leave the starch solution and amylase solution for 5 minutes before mixing them? Okay, so the clue here is that we're going to leave behind the starch solution for 5 minutes before mixing them. We know that we put them inside the beaker here, which is at a temperature of 5 degrees Celsius. But when you put something in water, it doesn't immediately change that temperature to what you want. It takes some time. And that's why you leave it for 5 minutes to allow both solutions to reach 5 degrees Celsius. This is called a water bath. Yeah, that's right, Elise. Very good. Thank you all for all the likes. Table 3 shows the results of the investigation. What conclusion can be made about the effect of temperature on amylase activity between 20 degrees Celsius and 65 degrees Celsius? So we want to see what's happening here. Obviously, you can see that the time taken until iodine solution stays yellow-brown in minutes will go from 5 to 14. You have to actually say that. So you can say, as you increase the temperature, the time taken also increases. But you need to specify. Oh, actually, there's one drop here. So you can say, as temperature increases, the activity increases. However, at 35 degrees Celsius, the activity increases up to 35 degrees Celsius. How do I know that? Because it's getting faster. When the time is smaller, it means that when the time is smaller, that means the activity is higher. That's your optimum temperature there. After 35 degrees Celsius, the activity decreases, meaning it has denatured. It's proven because there's less activity, and you can see that because the time increases until eventually it does not become yellow at all. Explain the results at 5 degrees Celsius and at 80 degrees Celsius. So they want it at 5 and at 8. 80, not 8. So At 5 and 80, they both did not become yellow-brown. They did not change. So let's talk about that. At 5 degrees Celsius, the amylase have low kinetic energy. Therefore, there are fewer enzyme substrate collisions. You can even say successful collisions, like in chemistry. So if you have a low temperature, low rate of reaction because there's no kinetic energy or low kinetic energy. Hence, yellow-brown color is not seen as the iodine did not break down the starch. So that's at 5 degrees Celsius. Now we have at 80 degrees Celsius. We said before that after 35, which was the optimum, it has denatured. So that's what you talk about. Yes, so they no longer fit together. That's right. So at 80 degrees Celsius, the amylase has been denatured. So the substrate no longer fits into the active site. The better way of saying that is that they're no longer complementary. That's always the best way of saying it. Hence, no starch were broken down. That's it. 5.8. The student investigated the effective temperature on amylase activity. Describe how the student could extend the investigation to determine the effect of a different factor on amylase activity. So we've seen temperature already. What other factors can we have? Usually it would be pH. So the pH is something we can say here. So temperature is now kept constant. Using a water bath. Always water bath. Change the pH of the solution. At pH 1 to 14 using acids and bases. There you go. Super simple. That's all you need to say. One of them should always be kept constant and the rest of them will be changed. Not the rest of them. One other thing should be changed. And that one of the other thing is the pH. You need to say that you're changing it from pH of something to something or concentration of something to something to show that you're using a range of values here. Then we have question 6. Which cell is the most transparent? Okay, I'd like you guys to answer this one. What would you say? Yep, oops, sorry. I'm very sorry. Let me just fix that for a second. Yeah, you guys are right. It is A. Transparent meaning that light can easily pass through. And the reason why it's A is because the chloroplasts are at the top here. So the light needs to reach the chloroplast. And to do that, the top must be transparent. So it's A. Which cell structure in the leaf mesophyll cell is not found in a root hair cell? What would you guys say is the answer for this one? Chloroplast, yeah. That's because the root hair cell, I can't spell, chloroplast, there you go. Because the root hair cell does not need any chloroplast, it's underneath the ground. It's not the nucleus because it's still a plant cell, so all plant cells has a nucleus. All animal cells also have a nucleus. The only one that doesn't is the red blood cell. Plants lose water through their leaves. Name the cells in the leaf that control the rate of water loss. Guard cells, that's right. The guard cells guard the stomata. The stomata opens and closes. It's literally the gap that opens and closes. And that guard cell, if it opens the stomata, then the water will leave by transpiration. No, you're not dumb. You're completely fine. I understand why you said cell membrane. But they did ask name the cells in a leaf, not the cell structure. Okay, which scientific term describes this movement of water? Yeah, transpiration. Transpiration stream. That's the correct term. Osmosis is the process of water coming into cells, but this is the process of water leaving the plant, and that's called transpiration stream. Which change would decrease the rate of water loss from a plant's leaves? So we want to decrease the rate of water loss. This is higher. Increase humidity. Very good. Okay. We'll go through 6.6 as the last one because I have a student after this. But at, what time is that? At 6 p.m. we'll do Gcse maths. So hopefully I'll see you guys there. Compare the structure and function of xylem tissue and phloem tissue. Okay, so xylem, firstly, it carries water and mineral ions. Phloem carries sugars. Xylem is made of lignin, and phloem have pores. Yeah. So you can say xylem is made up of lignin, hence made of dead cells, while phloem is made of living cells. The phloem requires living cells because it uses active transport. So that's something you can say as well. But let's talk about the structure first. So after that, we also have phloem cells have pores. While xylem cells do not. Xylem is hollow. That's why it's made of dead cells. While phloem contains cytoplasm. And then now you can talk about their function. So you can say, I think someone sent a GIF. Oh, a lot of you did. Thank you, Moonzy. So we have here xylem transports water and mineral ions, while phloem, I can't spell phloem, there we go, transports dissolved sugars. Xylem is involved in transpiration, while phloem does not. And my favorite one, xylem transports material in one direction. This one's the most common answer, while phloem is bidirectional, meaning it goes in two directions. Phloem goes both ways, that's right. The similarity is that both transport liquids through the plant. Okay, we'll end there for today.